So, good morning, everyone. Welcome to day 2 of our dose optimization workshop. I hope you enjoyed the dinner last night and slept all well and are ready for day 2. Welcome also to session 4. The title of the session is Dose Optimization in Early Development Options for the Path Forward. So we expect today another very intensive discussion, and we have great speakers invited. Today's overview is basically here on this slide. This session is going to last until 12:15. And then you are, of course, also invited to attend session 5, the last session of today, where we try to wrap up the entire conference, the workshop, highlighting the key conclusions and learnings, and then we close the meeting. I'd also remind you again, especially online, that you can follow us on media and Umbrella. And please use Umbrella and Slido also for the questions that you can submit online, so that we can have a very interactive and productive discussion in the panel. And with that, I'd like to introduce session 4, and this gives me the great pleasure to introduce my co-chair here, Chun-Se Lee from Roche Genentech. She's the executive director and distinguished scientist and head of oncology clinical pharmacology at Genentech. She is overseeing all strategic clinical pharmacology activities at Genentech at GRED for the oncology portfolio. And well, I was introduced yesterday already, so I think we can move forward with topic number 1. Chun-Se is going to talk about how to optimize the trade-offs.
Thank you, Harold. Good morning and good afternoon, everyone. Thank you, Harold, for the nice introduction. So it is my great pleasure to have this opportunity to share with you oncology dose optimization, how to optimize the trade-offs. So first, I want to briefly touch upon the translational oncology dose finding, the MTD paradigms. I think many of you already hear some of the speakers from yesterday on these paradigms, so which is including dose escalation and dose expansion. And the goal of the dose escalation for the MTD paradigm to finding the MTD and the DLT are the main driver for the dose findings. And additionally, I try to look at the laser. And once we identify the MTD, this molecule, the dose will further expanded into multiple indication and the combinations. So I think we also know, all the speakers already mentioned, the MTD paradigms works reasonably well for chemotherapy because the efficacy and the safety are usually highly correlated due to the mechanism actions of the chemotherapy, namely non-specific killing of highly proliferating normal and tumor cells. And however, these MTD paradigms may not necessarily applicable for the modern target and the immuno-oncology therapies. And here I just hypothetically drew the dose efficacy, which is blue, and the dose safety relationship. As you can see, the higher dose does not necessarily always translate better efficacy and a greater anti-tumor effect. And in the contrast, the higher dose may bring additional unnecessary toxicities. As a result, for this type of molecule, the biological effective dose or optimal biological dose may be more relevant for this type of therapies. And the other thing I also want to highlight, in reality, we don't know the true dose-response relationship curve. One of our tasks in the first-in-human studies is to characterize these dose-response curves, which later on we'll touch upon a little bit later. So what is the path forward for the oncology dose optimization? A holistic but not necessarily one-size-fits-all approach. So we need to select oncology dose in the overall context of the strategic context, namely what are the patient population and the disease-specific need, and how confident we are with the mechanism actions, and also CMC feasibility and medical need, etc. And under this strategic context, we have three important pillars. So the first pillar is innovative, adaptive study design, which is with the goal to collect robust clinical data across multiple dose levels to enable us to characterize the dose-response relationship and finding the right dose. And this approach can be including multiple approach or combination of the field. For example, backfill cohort, multiple dose expansion, and randomized dose finding, etc. And the second pillar is a full spectrum of data and knowledge. We can leverage preclinical and translational evidence and the PD biomarker, receptor occupancy, as the speaker has been mentioned yesterday. We can continue leverage clinical safety, efficacy, and PRO data to support those selections. The third pillar is predictive modeling, model-informed drug development. So again, as one of the speakers already mentioned yesterday, we can leverage some of the quantitative tools, such as dose exposure, response analysis, and PK/PD analysis to provide quantitative evidence to select the dose. Additionally, I also want to emphasize from yesterday's speaker, Woolley, for example, mentioned some statistics and AI machine learning model to try to address patient heterogeneities across different doses. So together, by leverage all these different tools, we can integrate all the data and the evidence together to support those selections. So first, I want to briefly touch upon the study design. It's more like a new concept of the mindset shifting. And instead of called dose escalation, dose expansion, I rename it as dose ranging, because the goal for the dose ranging is no longer finding the MTD, is to define the dose response curve for both safety and efficacy. Because here I also want to emphasize efficacy. That's also relevant yesterday's discussion. What are the patient population we would like to select for the first-in-human studies? If our goal is to characterize those response curve, we need to find the patient population is likely to respond to these molecules. And additionally, once we find those response curves, we need to leverage totality evidence to select dose or a range of the dose to be further defined in the dose finding studies using randomized fashions. Again, I also want to emphasize, if totality evidence data to show one dose is bring the benefit risk profile, we don't always need to do randomized dose finding. But most of the case, given limited information available in the dose ranging studies, we may not necessarily for sure to know that's the dose. So maybe a range of those may potentially work. That's the reason we do the randomized dose findings. I also want to briefly touch upon biomarker, which has been extensively discussed in yesterday. Again, clinical relevant PD biomarker is important to support dose optimization. And here at least a range of the biomarkers available in the clinical trial, ranging from target engagement, receptor occupancy, and the proximal biomarkers such as cytokine release or phosphorylation of downstream proteins, and also distal biomarkers such as CTDA, PSA for prostate cancer, CA125 for ovarian cancer. And lastly is the tumor shrinkage, the tumor size reduction over the time. So the first two, the target engagement and the proximal biomarker, are more relevant for the proof of mechanisms. So you can see in general, this biomarker more likely correlate with dose and exposure. And on the other hand, for the distal biomarker and the tumor shrinkage is the proof of concept biomarkers. And these biomarkers are a little bit far away from the dosing schedule, but it's more relevant for the clinical efficacy. So therefore, when we select a relevant biomarker to inform the dose, we need to take consideration of these nuances of these biomarkers. Lastly, I also want to emphasize biomarker is truly important for the support dose selections. But sometimes it can be misleading if we use biomarker in isolation without considering the context of the efficacy and the safety. I also want to introduce the quantitative pharmacology tools. Many of the speakers from yesterday also highlight some of the tools, which is including QSP modeling, population PK, PD modeling, and also those exposure response analysis. And in the panel discussion, we also discussed the PPPK modeling, try to understand the set of action of the exposure, and also potentially address the dose stratification for specific population. Additionally, we can also leverage meta-analysis and also platform predict disease modeling to inform dose selection. As Mark Ratner mentioned yesterday, Renee Bruno actually demonstrated by using tumor dynamic model, and we have been shown that the tumor dynamics regrowth are correlated better with overall survival, such as OS. So this information may potentially help us think about what are the efficacy endpoints we should use, to support those selections. I also want to introduce another tool, is dosing snapshot. I think Stacy yesterday also mentioned this dosing snapshot. And here I just a copy of that dosing snapshot that I guided from Friend of Cancer Research white paper as appendix. So the dosing snapshot is a template-based, with question-based template to summarize key information that are critical for the rationale of those selections. So which is including translational evidence, PK characteristics, safety and efficacy summary. And again, for the safety efficacy summary, we can include in biomarker relevant safety efficacy summary also. So in general, we can use this dosing snapshot as a communication tools or use as a living document to engage with internal cross-functional teams to prospectively discuss what's the dose finding strategy for this molecule. And also it's a powerful tool to engage external stakeholders, such as health authorities, to discuss dose optimization strategy across different stages of the development. As Stacy already mentioned, that FDA currently piloting a version of the dosing snapshot. They call dosing toolkits to optimize the communication between the sponsor and of FDA reviewer on the dose optimization for the drug granted as a breakthrough therapy designation. And I also want to mention that oncology dose optimization is not one-time thing. Should be seamlessly integrated into the early of the TPP and the CDP, which is target product profiles and the clinical development, and also cross the cycle of the planning. So we should start to think about the oncology dose optimization, dose finding strategy as early as the time preclinical candidate selection. And we can continue revise and fine-tune the dose optimization strategy once we have more and more data. So once we find the optimal dose or appropriate dose for the first indication, dose optimization is not end here, as we discussed yesterday. We need to also start to think about what's the dose finding strategy for new indication and in combination by leveraging the available understanding of the pharmacology of the molecule and the disease biologies. So I think lastly, I want to philosophically discuss what and how to optimize the dose. Actually, it's art. And we all know the oncology dose optimization is complex and multifactorial problems. So first is what to optimize. I think, the endpoints that we would like to optimize in the context of dose selection is efficacy and safety. And due to the nature of the phase 1 and phase 2 study, it's not possible to optimize the long-term survival as one of the efficacy endpoints. Instead, we need to rely on the early efficacy endpoint, such as ORR, CR, or the tumor dynamic metrics that are related, like Mark Ritton mentioned, the time to tumor regrowth, and also the regrowth rate, etc. Additionally, we can also leverage clinical relevant biomarker to support efficacy readout. And from safety endpoint perspective, we need to look into both acute and chronic toxicities, and also tolerability, such as PRO, and also dose intensity to reflect the tolerabilities. I also want to briefly discuss when to optimize. And again, the scope is a little bit different because, as I mentioned earlier, dose optimization strategy should be implemented across the entirety of the clinical development. So here, the one is more referred to randomize those findings? Should we do that, randomize those findings before or after the approval activities? So here, again, is my personal opinion. In general, as a default, I think we should do that after the approval of activity to show some anti-tumor effect. But again, this one also depends on the overall strategic context, how confident we are with the mechanism of action of this molecule for these pathways by leveraging, what's the competitive data is out there, and whether they already achieve POC. And also the prioritization for the entire portfolio for your company, how much resources and the money we want to put in, how important the speed in the context of unmet medical need, and also the patient competitions. Lastly, I also want to highlight, dose selection is always leave certain level uncertainties. As a human nature, we are not comfortable uncertainties, and also it's quite subjective. So here, I think here I just want to highlight another third point is the different stakeholders. So because different stakeholder will have slightly different flavor, different perception, what the level uncertainties we would like to accept. For some stakeholders, we may feel, we really worry about the efficacy. They tend to choose the higher dose to make sure we don't leave the efficacy on the table. On the other hand, for the other stakeholders, they may more emphasize tolerabilities. So they would rather pick a lower dose so that we don't make sure the patient is tolerable, willing to leave some of the efficacy out. So I think there's no right and wrong. So I think there need really closely collaboration and communication among all the stakeholders, among the patient, investigators, pharmaceutical industries, health authorities, and the payers, because all our intention is to bring transformative drug to the patient. So we have same intention, same goals. So I think the goal here is to further enhance the collaboration and the communication among all the stakeholders so that we can align the dose finding strategy without further delaying the oncology drug development. So with that, I would like to introduce the four presentations for today's talk. So the option for path forward. Hopefully by the end of the section, we can understand, we can leverage all these tools, the innovative drug design, full spectrum data knowledge, and the quantitative approach, and to figure out the path forward to identify the right dose for the right patient populations. So with that, I would like to acknowledge the Roche Genentech colleagues for their great contribution for this important topic, and thank you for your attention.
Thank you very much, Junzi, for a great presentation, very comprehensive, very informative, and staying within the time. So fantastic. You introduced already the next topics as well of this session. And for the next speaker, I would like to introduce Sophie Postel-Vinet, who is joining us online. Sophie is currently a clinician scientist at the Institut Gustave Roussy in Paris, where she is the senior medical oncologist in the drug development department, the phase 1 unit. And she's also the research group leader for Project U981 in CERM, the research unit, and the CERIC director. She became the head of the DTAP committee between 2020 and 2022, where she is the principal investigator of several early clinical studies. She's very much involved in early clinical research methodology discussions and topics, as well as in preclinical and translational research. In 2023, she started also to lead the institutional 5-year grant and label CERIC, the Integrated Center for Cancer Research. She is a member of ESMO, AACR, ASCO, and EACR, and she is involved in the organization or a member of the scientific committee of various international congresses. So it's a great pleasure to welcome Sophie online. And Sophie, the floor is yours.
So thanks for inviting me today to this Cancer Drug Development Forum. So I'm going to talk about integrating safety information from beyond the first treatment cycle in phase 1 trials. So these are my financial disclosures. And just as a reminder, you probably already have seen this slide many times, but just in a conventional chemotherapy, you have a linear relationship between the dose and the toxicity, and a quite close relationship between the dose and the anti-tumor activity. And therefore, the therapeutic index, which is between the minimal active dose and the maximal tolerated dose, is quite narrow. And this is where we define the recommended phase 2 dose. By contrast, with molecularly targeted agents and immunotherapy, you can see anti-tumor activity at much lower doses, and also the relationship between the dose and the toxicity is not always linear. Therefore, you have a broader therapeutic index where we can define a recommended phase 2 dose, but also recommended phase 2 dose range potentially for some agents. Again, just as a reminder of the definition of the MTD and recommended phase 2 dose as it has been done with conventional chemotherapy. Maximal tolerated dose is defined by the rate of observed dose-limiting toxicities, usually between 20 and 30%, which are per definition toxicities observed at cycle 1. And usually these toxicities are mostly grade 3 to 5 toxicities. By contrast, the recommended phase 2 dose is guided by the MTD, but it also takes into account the toxicities observed beyond cycle 1. So therefore, it's usually inferior or equal to the MTD. And this model was developed in the era of conventional chemotherapy, which was administered for a predefinite number of cycles. But by contrast, the current therapies, being either targeted therapies or immune therapies, are administered chronically, and therefore, the prolonged moderate toxicities or the late toxicities that we could call chronic toxicities, which occur after the first cycle, may become dose limiting, although they are not severe. As an example, here you have a grade 2 folliculitis with EGFR inhibitor or grade 2 hand-foot syndrome reaction with sorafenib. And this can be very debilitating for the patient. Again, as an example, some GI toxicities, such as grade 2 diarrhea, which is an increase of 4 to 6 stools a day over the baseline, can be extremely debilitating for patients. And therefore, we can really ask the questions of, are these toxicities tolerable? And are they going to have an effect on compliance? Now, to illustrate this also with immune therapies, this is the time to the toxicities, immune-related toxicities driven by ipilimumab. And as you can see, these clearly start to occur after the end of the DLT period. So this was really very nicely recently questioned in this article in the New England Journal of Medicine, which asked whether we should always go for the highest dose or selecting the dose for subsequent trials beyond phase 1. So to explore this impact of the late toxicities on the dose intensity, we initially performed a pilot study. This pilot study evaluated all the toxicities on trial, either skin toxicities, GI, or renal toxicities, in 445 patients included in 2 centers in 36 different trials. And in this pilot study, we observed that more than half of the grade 3-4 toxicities and more than half of the worst toxicities occurred after the end of the first cycle, that is, after the end of the DLT period. And therefore, we suggested to consider an acute DLT definition versus a chronic DLT definition. But because this came from a small data set, from only 2 centers, and with some specific data and not all the complete data from the trial, we created a task force, which was called DLT Target, which was created thanks to the EORTC, with the aim of defining new methods for determining dose-limiting toxicities and recommended dose for molecularly targeted agents. And we have a specific focus on severity and grade of toxicities, on the number of cycles to be considered, and on the relative dose intensity. So this is an overview of the patients and methods. And as you can see, it was a comprehensive analysis of more than 2,000 patients enrolled in 54 studies, which evaluated mostly small molecules, but also antibodies. The database also comprised more than 25,000 adverse events occurring in these 2,000 patients, which were reported during cycle 1 to 6 on trial, were at least possibly related to the drug and were not present at the same or higher grade at entry. So overall, the MTD was reached in 43 trials and the recommended phase 2 dose defined in 40 trials. Also, nearly 8% of the patients experienced DLTs. So this graph represents the worst adverse event grade over time. And as you can see, there is a significant proportion of grade 3 for adverse events, which are observed beyond cycle 1. As you can see, similarly, there is a constant and important proportion, nearly 30%, of grade 2 adverse events which occur beyond cycle 1. And this is currently not constantly reported in phase 1 trials. Now, with regards to the time to the first time to the worst toxicity. As you can see, 50% of the patients with grade 5 toxicity presented their first grade 3 or higher toxicity after the first cycle. And similarly, half of the worst grade 2 toxicities occurred after the first cycle. Now, this graph represents the adverse events of any grade over time. And each small bubble with a different color represents the toxicity which occur at cycle 1 for the white bubble, at cycle 2 for the orange bubble, and at cycle 3 to 6 for the blue bubble. And as you can clearly see, for some toxicities, the proportion of patients that present the toxicity of any grade is relatively constant over time. But for other toxicities, such as fatigue, diarrhea, or anorexia, the proportion of patients presenting such toxicities is higher at the late cycles, that is cycle 3 to 6. So now using the same color code and looking only at the grade 2 adverse event. What we can clearly see is that again, for some toxicities, such as fatigue, diarrhea, and neutropenia, the proportion of patients presenting such toxicity is higher at cycle 3 to 6, as opposed to the first cycles 1 and 2. And now looking at the grade 3 adverse event, again, we can see that some specific toxicities, such as diarrhea and neutropenia, are higher at the later cycles. So now looking at the relative dose intensity defined by the ratio between the theoretical dose and the effective dose administered per cycle. What we can see is that at any time on trial, you have at least 15% of the patients who do not receive the full dose intensity and receive less than 75% of the relative dose intensity. And if we specifically zoom at fatigue, for example, and relative dose intensity at cycle 1, what we can observe is that you have a proportion of patients which present grade 1 or grade 2 fatigue, which did not receive the full dose. So also the causality here could not formally be established. It suggests that sometimes not severe toxicities can be dose limiting even at cycle 1. So this led us to propose a model in which we would distinguish between the acute DLTs and the chronic DLTs. The acute DLTs would be defined by cycle 1 only and would determine the stopping rules for the dose escalation in order not to delay the trial and to be able to dose escalate rapidly. This would encompass severe toxicities at cycle 1 and some intolerable grade 2 toxicities. By contrast, the chronic DLTs would be either grade 3-4, but also intolerable grade 2 toxicities or repeated toxicities occurring at later cycles, and to which we could attribute a coefficient in order to weight them against the acute DLTs. Then to recommend the phase 2 dose, of course, we would need to include the PK/PD data, and then we would have two situations. Either the acute DLT is equal to the chronic DLT, the dose defined by the acute DLT, is equal to the one defined by the chronic DLTs, and then we would go for a phase 2 trial with the recommended phase 2 dose based on these two DLTs. If by contrast these one are not equal, then we could go for a phase 1 expansion with 1 or 2 cohorts or for a randomized phase 2 trial. in order to explore several doses, 2 or even a dose range. A last thing that we need to explore is the perception of the tolerability by the patient versus by the physician. So this is now much better explored thanks to the use of patient-reported outcome. But also, at this stage, we performed a survey which used the toxicities from the NCI-CTC AEs that we distributed to 52 patients. And we asked them to evaluate the tolerability of grade 2 toxicities lasting more than 7 days at 3 times on trial: baseline, so pre-treatment, early on treatment, and at the end of their phase 1 trial. And results are summarized on this graph. And what you can clearly see is that for most toxicities of grade 2 lasting more than 7 days, you have more than 30% of the patient which deem them to be intolerable. Also, what you can observe is that for these GI toxicities here in orange, usually they are feared at the beginning of the trial, and this doesn't change over time. But by contrast, for some toxicities, such as nausea here, or some neurological toxicities here, the proportion of patients that fear having such toxicity and find them intolerable for more than 7 days, even at grade 2, is higher on study completion rather than at baseline. And interestingly, when we distributed the same questionnaire to investigators, we could find that for neurological toxicities, the results were quite consistent with the one of the patients. But for GI or skin toxicities, the patients found intolerable some toxicities that physicians thought would be tolerable. So as a conclusion, we can say that in phase 1 trials of molecularly targeted agents, more than 50% of patients with grade 5 toxicities present their first event after the first cycle. A significant proportion of patients require dose reduction for selected grade 2 toxicities or even lower as early as cycle 1. And the collaboration with industry is, of course, key to build meaningful databases because as investigators, we only have a partial view on the data of the trial. So as main recommendations, in terms of dose escalation, the timing should be based on data from cycle 1 only, and dose increment recommendation should consider all available information, including late toxicities, which is currently done with the most recent trial design. All toxicities should be reported comprehensively, even if they do not occur during the DLT period in manuscript reports, and the recommended dose should incorporate all available information, notably toxicities observed after cycle 1, and should be based on achieving at least 75% of the relative dose intensity. So, of course, there are perspectives coming from these trials and also the new MDICT recommendation that were published this year. We should endeavor for having flexible design, which allow intra-patient dose escalation, backfilling, etc. We should potentially open criteria in order to allow enrolling specific population and determining specifically the dose for patients who have some specific organ deficiencies, and also could confirm the dose through randomized trials. Of course, it's key to implement the patient-reported outcomes in order to better evaluate the tolerability in real life, and also consider specific designs for combinations. And finally, it's important to involve regulatory authorities early in order to best define the right dose for these new oncology drugs. So with that, I would like to thank all these people who have contributed to this work, and of course, all the patients who participated to these studies and their families. And I thank you for your attention, and I'm happy to take any questions.
So really nice, it's excellent presentation, Sophie. So now it's our great pleasure to introduce Chad to talk about the PRO data. So Chad is the president and the principal consultant of the Gautany Consulting. He developed an innovative methodology to measure patient-centric outcome in clinical trials. He has published over 100 peer-reviewed articles and book chapters to address clinical outcome assessment and how the patient perspective can be examined to better understand the medical product and efficacy and safety. He also served as U.S. NIH grant to review committee and has co-authored many of the industry best practice guidance on development and the use of clinical outcome assessment. So Chad, with that, the floor is yours.
Well, thank you. My name is Chad Gwaltney, and I'm the president and principal consultant at Gwaltney Consulting. My consulting practice is focused on the measurement of patient experiences in clinical trials, and it is my pleasure to speak with you today about measuring and integrating patient-reported outcomes into oncology dose optimization studies. PROs are defined by the US FDA as a measurement based on a report that comes from the patient about the status of a patient's health condition without amendment or interpretation of the patient's report by a clinician or anyone else. So PROs are already widely used in late-stage oncology studies as secondary and exploratory endpoints, and in some cases, even primary endpoints. And we often equate the idea of PRO measures with broad, sometimes generic measures of quality of life, and certainly these types of measures are often used in oncology clinical trials. However, measures of the core disease-related symptoms of the condition are also important and have been used as secondary endpoints in pivotal studies in areas like myelofibrosis and lung and prostate cancer, and even as co-primary endpoints in myelofibrosis. PRO measures of the treatment-related symptoms are also increasingly included in trials alongside clinician-reported measures of adverse events. Both types of specific PRO measures, those that may be used to evaluate disease-related symptoms to capture effectiveness, and measures of treatment-related symptoms to capture elements of safety, might be useful in dose optimization studies. And in recognition of the increasing focus on the patient's experience in oncology clinical trials, the FDA has published a guidance document on core PRO measures to include in cancer clinical trials. And the listing of core PROs includes measures evaluating both the effectiveness and the symptomatic toxicity profile of the investigational treatment. I included this slide to provide an example of a measure that is primarily used to measure disease-related symptoms. And this is the non-small cell lung cancer symptom assessment questionnaire, and it has been qualified as a measure of NSCLC symptoms by the FDA. It includes a small number of items focused on things like cough and chest pain that are directly related to the underlying condition. So primarily a measure of disease-related symptoms that's used in trials to capture effectiveness data. And these measures of disease-related symptoms are important, but the primary PRO focus in dose optimization studies may ultimately be on the measurement of symptomatic toxicities from the patient's perspective, as indicated in this passage on screen, talking about adding PROs to clinician-reported adverse events in early cancer drug trials to better inform dose selection and optimization. So accordingly, in this talk, I'm going to spend more time on measuring this aspect of the patient's experience. So why do we care about measuring the patient's perspective on symptomatic toxicities when we already have clinician measures of symptomatic adverse events? Well, we know from previous research, primarily by Ethan Bosch and his colleagues, comparing the two sources of information, that clinicians may systematically underestimate the severity of the symptoms that patients are experiencing. And that phenomenon is displayed here, where the experience of fatigue, appetite loss, nausea, and vomiting is underreported in the clinician reports. So for this reason, measuring both the patient's perspective and the clinician's perspective on the symptoms that the patients are experiencing can be extremely useful. And this finding of a difference between clinician and patient reports directly led to the development of a PRO version of the CTCAE framework for classifying adverse events, which can be useful in evaluating symptomatic toxicities in dose optimization studies. And because of this potential utility, I wanted to spend some time walking through some details regarding its administration. So this item library that was created through the NIH in the US includes 124 items representing 78 symptomatic toxicities. So quite comprehensive, although still evolving and adding items as treatments with new mechanisms of action and potentially different types of toxicities are being tested. And these items capture a variety of different aspects of symptomatic toxicities, including how often they occur, how bad they are, how much they interfere with a patient's life. It is not required to use all of these items in every study. In fact, the idea is to select items from the library that are particularly important for a particular context of use. And I'll talk more about that in a second. And they're currently available in over 30 languages. And again, this continues to grow. On this slide, I have a listing of the types of symptoms that are included in the PRO CTCAE item library. And again, you can see that it is relatively comprehensive, including a variety of different organ systems and categories of side effects that patients might experience. And also this listing shows the different dimensions or attributes of each symptomatic toxicity that could be evaluated. Things again like frequency, severity, amount, interference, and just whether or not the symptom occurred. And then these are some examples of PRO CTCAE items. And I should have noted earlier that all of this information is available publicly on the NIH PRO CTCAE website. So much more thorough information is available. I just wanted to select some illustrative examples. So these items are asking about the symptoms that patients experience over the past 7 days. So we're capturing a 1-week time frame. There's no need to utilize a daily diary or anything like that in order to capture this experience over 7 days. The items are relatively simple and straightforward, as are the response scales. And this can be administered either as a pencil and paper assessment or electronically. So a variety of different options. And again, the idea is to select the items that are most relevant for a particular trial or program. So thinking operationally about how to implement these PRO CTCAE items in a specific clinical trial, the items are publicly available for use in clinical research. So if any of you have utilized PRO measures in previous studies, you know how challenging it can be to obtain licensing agreements and go through the translation process. It takes quite a bit of time to come to an agreement with copyright holders to utilize items. Much, if not all of that overhead is eliminated with the use of the PRO CTCAE, again, because of that they were designed to be publicly available. I've already mentioned that they can be administered on paper or electronically. That flexibility helps in implementing the measures in a trial. So one of the most common questions that I get about the PRO CTCAE is how do we use it alongside the clinician measures of adverse events? And the answer is it's not entirely clear at this point, but currently the most common practice is to report both. And so there's no need to harmonize the patient and clinician reports. We're not looking for differences between the two, and there's no need to come to an agreement about which one is correct, but rather the need is to present both so that there's a more comprehensive picture of what the patient may be experiencing. The items can be, as I mentioned, they have 7-day recall periods. So the idea is that they can be administered up to weekly. And this kind of weekly assessment might be particularly useful during key periods of the trial where there's a special interest in symptomatic toxicity and times when it may be particularly important to capture the patient's perspective on that. So again, one of the key aspects of using the PRO CTCAE item library is to select the items for a dose finding study that are particularly relevant for that particular product and in that particular context of use. There are a variety of different sources of information that could be useful, such as earlier stage research and the adverse events that have been reported above a certain level in a certain percentage of patients in that early stage research. If there are well-established adverse event profiles of compounds with similar mechanisms of action, that may be a source of information. The adverse event profiles of the most common treatments of a particular disease, given that new treatments may be combined with or tested against standard of care. Maybe from the patient's perspective, the adverse reactions that are of most concern to them may be the things that we want to focus on, or adverse reactions identified by expert stakeholder groups, or even animal studies may give us some sense of the types of symptomatic toxicities that should be included and measured through the PRO-CTCAE. So there's no one right way to do this, but rather involves the consolidation of a number of different resources in order to come to a final decision about a short list of items that could be included. Now, I've talked at length about the PRO-CTCAE, but there are other options to capture symptomatic toxicities using PRO measures. So there's the functional assessment of cancer therapies and the variety of different measures that are available in that battery of questionnaires. And even within that, there's a general fact measure where a simple single item about being bothered by side effects of treatment has been used as a kind of summary measure of how bad adverse reactions are from the patient's perspective. In the US, the MD Anderson Cancer Center has developed their own set of measures. EORTC in Europe has developed condition-specific measures, as well as a thorough, comprehensive item bank that can be used similarly to the PRO-CTCAE, where items can be selected that may be particularly relevant in a context of use. There are a number of different options that are available beyond the PRO-CTCAE, and it's up to the researchers to decide which one might be most appropriate for a particular dose optimization study. So in conclusion, patient-reported outcomes can be extremely useful in examining both effectiveness and maybe in this context with dose optimization, particularly of particular importance would be symptomatic toxicities in these dose selection studies. There are a wide variety of options that are available. The PRO-CTCAE may be one to consider in most situations, given that it's widely used and publicly available. And I think importantly, I didn't mention this earlier, but again, anyone who's implemented patient-reported outcomes in cancer studies knows that sometimes it can be very complicated to establish the validity of PRO measures in a way that meets industry or regulatory standards. With many of these items, including the PRO-CTCAE, they've been tested already, and therefore, in many cases, will not require additional validation, which I'm sure would be welcome news to many of the study teams, where, in particular, in early stage dose optimization studies, where there just really is not time or the resources needed to do that kind of testing with PRO items. And then finally, I'm keenly aware that the title of my study was, or my talk is about integrating patient-reported outcomes. I think I've spoken more about implementing rather than integrating. And the straightforward reason for that is that the processes for including these kinds of PRO measures are easier to articulate than determining how to use them in concert with other sources of data and other endpoints to make dose selection decisions. And I think that's one area where the field will need to advance in order to decide how do we, not necessarily algorithmically, but how do we put all this information together to make a decision? Right now, I think it's a lot of information sitting side by side with a lot of value in that the PRO measures provide a window into what the patients are experiencing, and that source of information is helpful in making decisions, even if the process for making that decision is not well established at this point. And with that, I'd just like to say thank you to everyone, and particularly to the meeting organizers for giving me the opportunity to speak to this important topic, and to participate remotely, given that it's quite early right now where I'm located on the East Coast in the US. So thank you very much.
So I think we're up to the time. So if you have questions, feel free to reach out to speakers. And now we can go to the coffee break. And please come back at 11:45. And we will start the section sharp. Yeah. Thank you, everyone.
We have changed the program slightly because we realized that after session 4, we hadn't included a Q&A session. And as questions are coming in already from the online participants, we will have a 15-minute Q&A, a panel discussion at the end of session 4. And yeah, now it's my pleasure to introduce our next speaker. Our next speaker is Johan de Bono, who is providing us with the academic perspective, including some case studies on the topic of dose optimizations and options for the path forward. Johan de Bono is the Regius Professor of Cancer Research at the Institute of Cancer Research and the Royal Marsden Hospital, and he is in the lead, or was in the lead of the development of abiraterone, cabazitaxel, enzalutamide, olaparib, lutetium PSMA, and the molecular genomic stratification and germline sequencing of advanced prostate cancer. I think Professor de Bono is a very well-known expert. He is leading the drug development unit at the Royal Marsden Hospital, comprising of more than 100 people, staff, and has been conducting more than 50 phase 1 clinical trials at any one time, and is running a large research laboratory in addition. So again, great pleasure to introduce Professor de Bono and invite him to start with his lecture.
Hello, my name is Johan de Bono. I want to talk to you today about the title I was given for this talk, which is Some Case Studies on Dose Optimization. And I want to talk particularly about 3 drugs whose drug development I've been very involved in over really 20 years that have led to drug approvals and that changed patient care, but have had drug dose optimization questions really analyzed in detail in these studies. So here's my talk overview. I'm going to start off, first of all, with the introduction. So arguably, variability in drug PK is diminishing as drug discovery efforts are focusing on developing agents with lower or limited PK variability, limited CYP liabilities. And this is especially, I guess, relevant because biologics overall have lower PK variability, for example, antibody treatments. However, despite this, cancer's complexities makes drug dose selection challenging. And I'm going to talk about this a bit more in this slide and the next slide. Drug delivery to tumor cells remains a challenge. Tumor heterogeneity, both intra and interpatient, remains a challenge. And actually, the temporal, not only the spatial, but the temporal changes as the tumor cells adapt to drug dosing remain a challenge. And let's divide that up a bit further. And my overall premise is that recommending the same drug dose to all patients for all cancer types, indeed for all the cancer cells for a particular patient in different parts of that patient's body, in my opinion, is not always rational on what we know about cancer disease biology. So let's talk about drug delivery to tumor. Many years ago, we did trials with PET tracer labeled drugs. And what we learned is that actually delivery to cancer of drugs remains a big challenge. For example, in liver mets, we saw good distribution of drug in the peripheral zone of the tumor, but minimal drug distribution to the center of the tumor in work we published in JCO many years ago by Proper et al. So we have to be concerned that disease hypoxia, hydrostatic pressure in the tumor, poor blood flow, dense stromal reactions, for example, in pancreatic and colorectal cancer, limit drug delivery and remain a concern. And you can get delivery to some parts of the cancer that are very good, but not to other parts of the cancer. The other issue is disease heterogeneity. For example, an EGFR inhibitor may be much more active against a mutated EGFR versus a tumor that has EGFR amplified, EGFR hyperexpressed disease, or mutated AR and mutated, AR with a different mutation, for example, or AR amplification with very high AR expression, AR being the androgen receptor. So there are really variations in one cancer that are a challenge, and also between patients. And not only in tumor genomics, but also in epigenetics and tumor stroma between patients. Moreover, the cancer adapts following treatment, and direct drug target perturbation can alter that target by new mutations, increased expression. For example, AR becomes overexpressed following therapy very quickly often, which may actually mean you need a higher drug dose over time in the same patient to actually can keep on hitting target. Maybe even the tumor chemoattracts myeloid cells or alters other stromal cells in prostate cancer and fuel the androgen receptor signaling over time. So you may need more drug to hit targets. So let me give you some examples of these challenges in drug development dose optimization. I'm going to start with cabazitaxel, a drug that was specifically made by Ron Plonk many years ago as an agent that would work when paclitaxel and docetaxel were not working. So this reverse resistance. There was 2 phase 1 trials with this drug run by Ron Plonk. I was involved in that phase 1 study when I was working in San Antonio as a clinical fellow. And the 2 trials recommended 2 different doses. One was recommended phase 2 dose of 20, and the other one was 25 mg per meter squared. And then a breast cancer trial actually pursued a phase 2 trial that actually started at 20 mg per meter squared and actually recommended dose escalation in patients that tolerated 20 mg per meter squared to 25 mg per meter squared. Really, dose escalation based on tolerability that is unusual in cancer medicine. And in the breast study, we could see that some patients really were undertreated at 20 mg per meter squared, and some were overtreated at 20 mg per meter squared in view of a CYP3A4, metabolized very, drug that has variable PK. Now, what happened with this drug was that Ron Plonk became Sanofi against Sanofi Aventis. There was some loss of memory in the company, and I had submitted an LOI for randomized phase 2 study in CRPC, and really this drug was somewhat forgotten until really docetaxel was coming off patent when cabazitaxel was revisited, and this drug was taken straight to a phase 3 trial with minimal, really phase 2, in fact none, efficacy data. And the dose taken forward to the phase 3s was 25 mg per meter squared, Q21 days, based on the premise this was going to be more active probably, but perhaps more toxic. And indeed, that was the case. We have shown with this drug, as you can see top right, a survival benefit. The drug got approved by the FDA, and then the FDA mandated 2 post-registration trials and specified their precise designs, looking at 2 different doses of cabazitaxel, 20 and 25, and comparing these either against each other in the post-chemotherapy space in the Proselica trial, or against docetaxel in the first-line CRPC trial against docetaxel. And overall, not surprising, we showed that 20 mg per meter squared is better tolerated than 25, with less neutropenia and neutropenic sepsis, although those rates were low. But actually, we still know that actually cabazitaxel has a higher response rate at 25 mg per meter squared than at 20 mg per meter squared, although now 20 is the recommended dose. And the non-inferiority trial that was run as recommended by the FDA had broad confidence intervals. And actually, we still cannot say that 25 is the suboptimal dose, because actually the trial simply studied whether the reduced dose decreased OS by 15% or more in that non-inferiority trial design, and we actually rejected that hypothesis of a 15% reduction in survival benefit. So what have we learned from all that with that case with cabazitaxel? Well, we've learned that cabazitaxel improves outcome. But actually, we've also learned that the lower dose is better tolerated, but actually it did not decrease OS by by more than 15%. But actually, the higher dose does have more tumor activity, not surprisingly, and it's clear that some patients are underdosed and some overdosed. And in fact, in one patient, I'm confident that some tumor cells are getting good exposure and some are not. And perhaps the main lesson here is that oncologists need to consider dose escalation as well as dose reduction. Let me go to the next example, abiraterone. Abiraterone is a drug that was generated by chemists in my institution, the ICR, as a CYP17 inhibitor and blocks AR signaling. The first in human evaluation gave a short 14-day exposure, which confirmed target modulation and reasonable PK. And then we started trials that have demonstrated that the term hormone refractory prostate cancer is a misnomer. But interesting, in the study, we saw response from the very first phase 1 dose level of 20 to 50 mg per day continuously. The drug was very well tolerated. There was no DLT. And we saw target modulation at all dose levels. Now, the PK did demonstrate a moderately high PK variability, and the drug was given fasted. And we showed that the food actually decreased PK variability and increased bioavailability, although actually our colleagues in industry were not particularly keen really to change the recommended dose because the time was against them, and they were running with the drug trial to move to phase 3s very quickly, even though a formulation switch was required just before the phase 3 trial started. Now, this drug improved overall survival. Arguably, this drug has been one of the most impressive drugs in prostate cancer medicine ever, really, and is actually impactful not only in late-stage prostate cancer, but even from diagnosis, improving overall survival by at least a year and adding many hundreds of thousands of life years over the last decade. But actually, what remains the question is, what is the best dose and how best to give this drug? Perhaps we're giving it with the wrong steroid. In fact, we should probably give dexamethasone, not prednisone, because this is a better way of blocking the increased melanocorticoid steroids upstream of CYP17. Perhaps this drug is better used with food to minimize the toxicity of the higher dose, although I think those studies have never really been done well enough in a randomized setting. But certainly it's feasible to give the drug with food and decrease exposure. But the speed at which this drug was developed really limited optimizing the, I think, way this drug is given to cancer patients. Let's move on to olaparib. Olaparib is another drug that I've been very involved with from phase 1 through to phase 3. It was generated by Kudos, a small biotech in Cambridge, UK, founded by Professor Stephen Jackson. Work in the lab of Alan Ashford and Thomas Halliday showed that there was a synthetic lethal interaction between PARP inhibition and BRCA loss of function. And we set up a trial with olaparib, and I, with my team, decided to pursue a twice-daily continuous dosing regimen with the capsules of the drug. And even to date, I'm not sure the scheduled continuous dosing was optimal with a cytotoxic agent like olaparib. Anemia was dose limiting. We saw PD modulation at the very lowest dose levels. Really, even at 100 mg twice daily, we're seeing really good PD, 100% PD modulation. But there was still a clear dose-response relationship from that dose level where we had 100% PD modulation to the highest recommended dose of the drug, which was 600 mg twice a day with capsules, and that was a toxic dose. Now, again, like abiraterone, a formulation switch was required with this drug, and that made things more complicated. We had to switch from capsules to tablets, and the tablets at 300 mg per meter squared were roughly equivalent to 400 mg twice a day of capsule. So this trial moved to randomized phase 3 trials. This drug has changed the care of ovarian, breast, pancreas, and prostate cancer. These are the prostate cancer data out of the prostate cancer phase 3 trials. But there's increasing complexity with this drug. Different subtypes of prostate cancer have different sensitivities to this drug. BRCA2 loss, super sensitive. PALB2 loss, super sensitive. ATM loss, still sensitive, but less sensitive with biallelic loss. Not every gene is created equal. Moreover, there are different mechanisms of resistance. For example, the ABCB1 pump, the PGP pump, can cause resistance to this drug, and therefore higher doses may reverse resistance to lower doses. Moreover, there are other mechanisms of action muted, although not proven in clinical trials. For example, this drug is muted to inhibit AR signaling, although that's not really proven in the clinicals. We never really have seen PSA falls in patients without DNA repair defects, as we published in the England Journal of Medicine by Matteo et al. So what have we learned from olaparib? Well, it's clearly got anti-tumor activity against tumors with DNA repair defects, particularly the genomic BRCA2 HOMDELs, or biallelic loss with mutation and het loss of the other allele. But the detail matters. The type of genetic alteration, genomic alteration in a tumor, does make a difference to the level of sensitivity, a bit like the EGFR mutation, EGFR amplification story of EGFR wild type. Different EGFR mutations are very different for different drug sensitivities. But also olaparib, despite a dose level that was hitting target at the PD level at 100%, still had a clear dose response relationship above that maximal PD modulation dose level. And actually we saw further, dose response, suggesting that the higher dose is still important for this molecularly targeted synthetic lethal interaction, although clearly, the lower doses are less toxic. Moreover, different PARP inhibitors seem to have different off-target effects, and we have to be thinking with these drugs about their off-target impacts. And in particular, one of the main effects for these inhibitors is that they inhibit PARP1 and PARP2, and perhaps the PARP2 blockade may also cause some of this toxicity. And we now have PARP1 selective inhibitors that are sparing, it is thought, some of that hematological toxicity, and maybe by giving a higher dose, maybe more active with regards to anti-tumor activity. So what do we learn in conclusion? Optimal drug dosing remains a complex challenge that may not always be something we can address fully in a first-in-human trial. I think overall higher doses do have more anti-tumor activity generally, but are more toxic. And the optimal dose and schedule may vary from patient to patient with the same drug in the same disease, from site to site in the same patient, even over time in the same patient as treatment induces cancer adaptations, and between different diseases. So thank you for your attention, and I'm really looking forward to this meeting and the discussion we're going to have about this question. So thank you.
So next, really appreciate the nice, excellent presentation. Next, I would like to introduce the next speaker, Divya, and she will present on behalf of the IQ Consortium on the industry perspective with case examples. So Divya have 12 years industry experience in clinical drug development for small molecule, large molecule, and serve as a team leader and people management. She have lead clinical pharmacology strategy for multiple myeloma disease area strategy to support global development and cancer therapy. And she is also very passionate to drive science-based model-informed drug development to maximize benefit-risk profile and enhance access to anti-cancer drug therapies. So without further ado, I would like to let Divya to take the stage to share her stories.
Thank you, Chunzi, and I would like to thank the organizers here for having me here to present my thoughts on behalf of the IQ Dose Optimization Working Group. Please bear with me if some of the thoughts seem repetitive. That's the risk you run into when you're the last speaker. So before I begin my presentation today, I would like to acknowledge the contributions of Neeraj Gupta, who has tremendously aided in co-creating this presentation with me. And as I mentioned before, we are presenting this on behalf of the IQ CPLG Dose Optimization Working Group. So our working group started in Q4 2021, and one of our key objectives was to provide a recommendation for the dose optimization strategies for the oncology programs. So in order to gain insights on the current industry practices for oncology dose finding, we conducted a survey early last year, and some of the key components or the questions that we asked in a survey was, what are some of the strategic considerations in terms of dose selection? What are some of the feasibility challenges that the sponsors are facing in terms of implementing dose finding studies? And what is the impact of Project Optimus on dose optimization strategies within the sponsor companies? We had a decent turnout on the survey with 18-member companies responding to it. And in the next few slides, I'll show you some of the key insights from our survey. So this slide here shows the results on one of the questions we asked in our survey on the importance of various factors when it comes to informing an optimal dose. And some of these factors included a clear understanding of long-term safety and tolerability, evaluation of clinical dose exposure efficacy relationships, and mechanism-informed pharmacological contextualization to the clinical doses tested using evaluating the degree of target engagement using biomarkers in the surrogate tissues, verification of desired modulation of downstream pathways using biomarkers in tumor microenvironment, and translational evidence to contextualize human PK. And on the right side here of the slide, we are showing how often in practice do the sponsors have this information available. Sponsors agree that a clear understanding of long-term safety and tolerability, in addition to evaluation of dose exposure efficacy relationships being critical, and this information is often available in practice. When it comes to mechanistic understanding using biomarkers in surrogate tissues or tumor microenvironment, these are considered supportive. Interestingly, sponsors also noted that when it comes to biomarker information in the tumor microenvironment, this is not usually available to inform dose selection. And this may likely be due to operational constraints or maybe because sponsors don't have a proactive biomarker strategy. In the next slide here, these are the results from one of the other questions we asked on the feasibility challenges that the sponsors have been facing. So the most frequent ones being delays in market entry or delays in patient enrollment, lack of meaningful accessible biomarkers, as I mentioned earlier, was also one of the frequent reasons. Interestingly, sponsors also noted internal alignment issues, such as resistance to collecting clinical data from additional dose levels. So we are hoping Project Optimus could enhance or improve this cross-functional alignment. Sponsors have also noticed the lack of timely clinical trial data, such as PK biomarkers, and also time or resource constraints for incorporation of newer MNS technologies. So when it comes to dose selection in oncology, it's often a problem of multidimensional optimization. I don't know what just happened, bear with me. Some of the key dimensions that require optimization includes dose, schedule, line of therapy, cancer type, histology, patient selection, combination partner selection, dose and schedule of the combination partners. In one of the case studies today, we'll show you how tumor mutational status was one of the key dimensions to optimize the dose. Emerging biomarker technologies, integrated pharmacological frameworks such as QSP could enhance our ability to tackle this multidimensional optimization and also could lead to the development of optimized cancer therapeutics. So as expertly illustrated by Venkat Krishnan et al. In the latest CPT paper, a case-by-case approach is warranted to tackle this multidimensional optimization. So confidence in dose optimization should be supported by 3 interdependent anchors, as noted in black here. A robust mechanism-informed quantitative pharmacological contextualization as a foundational base, clinical efficacy as demonstrated by a clinically meaningful improvement in benefit, long-term safety tolerability characterized by low incidence of dose modifications and favorable patient experience. The key cultural enablers of success, as shown in green here, include comfort with an iterative learning approach towards multidimensional optimization, commitment to a totality of evidence approach, and interdisciplinary collaboration, also incorporating patient voice into the clinical trial design. The key technical enablers of success, as shown in orange here, some of those include experimental models of cancer biology, pharmacology, at emerging biomarker technologies, systems pharmacology models, pharmacometric models, evaluating a longitudinal assessment of clinical data for the endpoints such as efficacy, safety, tolerability, such as PROs, or enhanced clinical trial designs for that matter. Today I'll show you a few case studies that have effectively incorporated this totality of evidence approach to inform dose optimization. The first one being mosunetuzumab. In short, I'll call it MOSUN. It's a bispecific CD20, CD3 T-cell engager. It was granted accelerated approval and conditional marketing authorization in 2022 for the treatment of patients with relapsed refractory follicular lymphoma who were given 2 or more prior lines of systemic therapies. So it's a full-length humanized IgG1 antibody with one arm of the bispecific targeting the CD20 antigen on B cells, and the other arm targets the CD3 antigen on T cells. It acts as a conditional agonist. It works by dual binding of the construct to B cells and T cells. Upon binding, it results in the formation of an artificial immunological synapse, which leads to T cell activation followed by the release of cytotoxic granules such as granzyme perforin, leading to tumor cell death. CRS is a major safety concern for bispecifics and CAR-T therapies. It's considered as an on-target toxicity, So following the bispecific administration, the formation of an immune synapse leads to T-cell activation followed by an activation of immunological cascade, leading to a CRS risk. So pharmacologically, one can expect that as you increase the dose of the bispecific, there's a greater increase in target engagement, which can lead to a higher safety risk such as CRS. So a key developmental challenge when it comes to bispecifics is to design a dosing framework that mitigates the CRS risk while enabling the delivery of an effective dose. And one way to do that is to use a step dosing approach. So in order to understand the CRS risk, it's important to understand IL-6 and its kinetics. So patients with CRS at an average tend to have higher levels of IL-6. So it's a useful biomarker to draw inferences about CRS. So before the clinical data was available for mosunetuzumab, preclinical data along with data from the published literature was leveraged to inform the phase 1 study design for MOSUN and also to inform the design of a dosing strategy. So for this purpose, a systems pharmacology model was used by leveraging data from preclinical studies as well as the published literature that describes the T-cell activation and IL-6 release in some of the key physiological compartments. So for further details on the model development and application, please see the references below. So in order for MOSUN to work, it needs both T cells and B cells. So this model nicely describes the B cell generation and maturation in the bone marrow, its release and trafficking into the key tissue or physiological compartments. It also described the generation of T cells from thymus, which were released into which describes the release into the peripheral blood, as well as the trafficking to the other tissue compartments. So T cell activation is a function of MOSUN concentrations along with B cells and T cell numbers, and this is an upstream driver of IL-6 release. So this model was then used to predict the IL-6 release and compare different dosing scenarios for MOSUN. So on the right here, I'm showing 2 different dosing scenarios for illustration purposes only. So on the top right is a fixed dosing scenario where MOSUN was administered at 20 milligram dose on day 1 of every 21-day cycles. And in the bottom dosing scenario, the 20 milligram dose was preceded by a small step dose of 1.6 milligram administered 7 days prior to the 20 milligram dose. As you can see, the model, the systems pharmacology model, was predicting about a 0.5-fold reduction in the peak IL-6 levels with the step dosing regimen compared to the fixed dosing regimen. So now, in other words, the model is saying that you could actually get to an effective dose of 20 milligram, but with only half of the concentrations of that of the fixed dosing regimen when it comes to IL-6 levels, which is presumed to lower the CRS risk as well. So taken together, the systems pharmacology model informed using mechanistic insights helped design a dosing framework that, while not only mitigating the CRS risk, enabled the delivery of an effective dose, thus maximizing the therapeutic index. So these learnings are broadly applicable to other bispecifics as well. So this is another case study that nicely illustrates the pivotal role of quantitative pharmacology to inform dose optimization for a molecularly targeted precision medicine, asciminib, which is an allosteric inhibitor of BCR-ABL kinase in chronic myeloid leukemia chronic phase patients. So asciminib is active against both the wild type and mutant forms of BCR-ABL kinase, such as T359 mutations. So a mechanistic PK/PD model was developed in this case to describe the longitudinal time course of BCR-ABL transcripts using data from 300 patients from both phase 1 and phase 3 studies to characterize the exposure-response relationship while also confirming the proposed doses in both the wild type and the T359 mutation patients. In this case, it was a 3-compartment model with a dynamic interplay between proliferating cells, stem cells, and resistant cells. The covariate and the magnitude of drug effect was actually included on the proliferating cell compartment. The covariate effects showed that for patients with T359 mutations, they have more resistant cells. They're predicted to have more resistant cells, less proliferating cells, and a lower magnitude of anti-proliferative drug effect. So for further details on the model, please see the references below. So this model was then used to inform the exposure-response relationships by predicting the longitudinal time course of the BCR-ABL transcripts for patients with both the wild type and the T315I mutations. So on the top right is a longitudinal time course of the BCR-ABL transcripts across the various dosing regimen. So while the model shows that there appears to be an exposure-efficacy relationship, it did not translate into a meaningful improvement in the clinical benefit at both the 24 weeks and 48 weeks of dosing. But when it comes to the patients with T315I mutations, the model predicted a meaningful improvement in clinical benefit, with the highest improvement observed at the dose of 200 milligram BID. Importantly, the model showed that at the daily dose of 80 milligrams per day, the model predicted exposures with only less than 5% coverage above the EC90, but whereas with 200 milligram BID dosing, the model was predicting greater than 95% coverage of exposures above EC90. So taken together, the clinical safety data, the flat exposure-safety relationship, and the learnings from the PK/PD model, it provided a robust justification to inform the differential dosing for patients with underlying molecular pathology, thereby supporting a 5-fold higher dosing for patients with T315I mutations compared to the wild type. So viewed from a broader perspective, these two studies efficiently illustrate the application of a totality of evidence approach by leveraging clinical data across multiple data sets and integrating them in a mechanistic manner. And confidence can be obtained in dose optimization by consistency across multiple approaches. In summary, a holistic approach to examine the totality of evidence using clinical data, preclinical data, biomarker data, based on a comparative assessment of benefit risk across multiple dose levels allows for an efficient characterization of exposure-response relationships and subsequently informed dose optimization. When it comes to phase 1 data, the sample size is always limited. It can be highly heterogeneous. So any efficacy inferences one can make based on the phase 1 data can be limited or rather inconclusive. So project teams should make efforts to collect biomarker data based on pathway or disease modulation. And one way to do that would be either through multidisciplinary collaborations and or proactive biomarker strategy. In addition to evaluating long-term tolerabilities, project teams should consider incorporating PRO CTCAEs. And when incorporated in the phase 1 study, it incorporates patient voice when it comes to dose selection. Additionally, any exposure response analysis that can be done using PRO CTCAEs provides additional evidence to the safety and tolerability of the drug. and also use proactively and properly quantitative pharmacology tools such as systems pharmacologic models, pharmacometric approaches using longitudinal data can enable efficient integration of all the clinical data and could also allow to guide the dose selection, maximize the patient experience, and also improve the success of the drug development. And last but not least, and Chunzi nicely articulated it earlier this morning, oncology dose findings always operate with a certain level of uncertainty, and alignment on the level of uncertainty with health authorities is critical. While the dose snapshot tool that was discussed by several of our speakers, while it can facilitate both internal alignment as well as communication with health authorities, it can also allow earlier identification of dosing-related issues and also enable timely mitigation. With that said, I would like to thank the audience for your participation, and also I would like to acknowledge the contribution from our IQ dose optimization working group. With that, yeah, back to you, Chunzi. Yeah, thank you.
So now I would like to invite the speakers and also sit on the panel discussion. We have around 15 minutes Q&A, and also the speaker online. Yeah.
So maybe we can start with the first question that is coming from the remotely participating group. How do we take, this is I think a question for Sophie, how do we take into account early dropouts for evaluating late toxicities? How has this been factored in?
Basically, I'm not sure I get the question, but early dropouts, the patients that drop out of the trial were not considered when we looked at all the subsequent cycles. So every time a patient dropped out of the trial, he or she was censored when we did that analysis. So the number of patients analyzed was gradually decreasing over the number of cycles. So what I showed was the proportion, and it's true that the number of patients was gradually decreasing. Then I think it does not mean that effectively these patients also present acute toxicities, which have to be taken into account as well. But this is a completely different profile of toxicity. So I think if a patient goes out for toxicity, it's a different thing to be considered as whether it goes out just for disease progression. And it's true that this is not something that we distinguished when we did that analysis. We just censored the patient, not differently between going out for toxicity or progression.
Maybe I can comment as well. The problem with phase 1 is that most patients are off trial usually within the first 2 or 3 months. So basically, for most patients, defining late toxicity is very difficult because they're not on trial for very long. And this really relates back to what Chunzi said earlier in her talk, great talk, that actually we may have to wait until really we're in later stages of drug development when we know about the efficacy of the drug, before we can really invest in what has been called, perhaps imprecisely, optimization.
Thank you very much. We have a question here in the audience.
Thank you. I'm Philip Josephson, European regulator. I think several of the very good presentations today and also yesterday pointed out that one of the big challenges of what is meant by dose optimization. I guess when we hear that word, we think of dose optimization as having the right dose in the product information at the time of approval. But it's also extremely clear that one dose will not fit all. The optimal dose will vary with a number of parameters, and there's not going to be a single optimal dose. How do we, and I think we all recognize this, so how does that fact sort of fit into the larger scheme that we're talking about here with respect to the most, to the best way to sort of get to a labeled dose, including, of course, dose reduction schemes and similar things? If anyone who feels like they want to comment on this, please.
Maybe I can reply, Johan de Bono here. You know, I think we have to be humbly aware of how complex cancer is, and we should not use the word optimization, in my opinion. That is a misnomer. precise language does matter. We should instead use the term biologically active dose range at the end of a phase 1. You know, we can at least get our descriptors precise.
If I may add to what was being just said, abstract concepts for dose optimization may not be real. So it takes a lot of deliberate effort to get there, right? When it comes to, it's more about, as Chunzi nicely said, it's about understanding the exposure-response relationships and what is the confidence that we derive from it. For example, do we have a clear contextualization of the clinical doses that we are testing using all the relevant information, preclinical, clinical, using data from molecules with similar MOA? What does efficacy look like? Is there any meaningful improvement in benefit? Is there a clear long-term evidence of safety and tolerability? And also, it's also important to note that when it comes to dose optimization, what I mean by being abstract is not real is it's a program to program perspective, setting those clear quantitative targets very much early in development. For example, when it comes to safety, what are the clinical AEs of concern? What are the grade AEs of concern? And what is the difference in AE grade that is considered meaningful that warrants an evaluation of a lower dose? And could you find a delta when it comes to efficacy using the sample size that you have and the endpoints that you have? So it has to be, the project teams need to be comfortable with that iterative learning approach. Yeah.
And to add to that, you know, patients treated in phase 1 trials are much more robust and usually younger than patients treated in phase 2 and 3 trials, particularly phase 3 trials where you have, you know, 150 sites. Certainly for my disease, prostate cancer of interest, you know, that's definitely the case. And because of that, we're treating frailer patients in the later stage trials, and therefore the tolerability will differ. So, you know, to actually assume that even from the tolerability perspective, we can get it right at the end of the phase 1 may be, you know, challenging because the population will change when we move to phase 3.
I kind of agree with your comments. I think, you know, how we define the optimize as a term, actually, we probably never have the dose for each individual patient. So I think we just need to be practical. So given the patient heterogeneity and also the, you know, in the context of both efficacy and safety. So we need to be realized that we just want to find a appropriate range of the dose that may work for the majority of the patient. That's potentially our best shot at this moment. Of course, when we continue learning, get more and more experience, we can start to think about in the future, right? We can personalize to finding the right patient population to give their personalized dosing. But unfortunately, in the drug development, we need to learn, collect the data. Eventually, we get there. Yeah, we're still not there yet. Yeah.
Agree.
Yeah, I don't know whether it's a question, Stefan Braun speaking from Zug, or more a comment. Let's see. So let's assume we get everything right in the preclinical setting, right? We enter the human phase and there will be surprises. We have a very well-established system that uses DLTs. And now we're coming up and say, let's question it. It's probably accurate, right? We're all, at least the physicians, entered the hospitals to treat patients. So now let's listen to the patients. But what we see now is they tell us a completely different story. How do we use that information now? So the FDA says, let's use it. Okay, let's use it. But do we report a grade 4 patient-reported outcome? Or do we ignore it? But we assess it. So what is the next step here, right? So obviously we need to get the dose right, but if you think it through, the best thing probably you can do in the clinic is you have a dose and you de-escalate. I have not seen a lot of successful drugs where you say, let's escalate, right? That's difficult. So how do we integrate that now? I think it's interesting information. And I'm intrigued to use the PRO CTCAE, but what do I do with it, right? Is that relevant information or is it just disruption? Telling me the sites that I have selected and trust in don't do the proper job because the patient is telling a different story. So all that is, as usual in science, right? You ask a question, get multiple answers. It's more confusing than before, even though you thought you have identified an issue. So any thoughts from my panel and presenters? By the way, thanks for great presentations on this. I'm getting a little bit confused now.
Who wants to take that question?
Hi, this is Chad. I can maybe start. And unfortunately, I don't think I have a great answer for that. In fact, in my talk, I mentioned, I think we know much more about implementation of PROs to capture symptomatic toxicities and tolerability than we do about how to truly integrate them with the other sources of information that we have to make decisions. So I think the best practice right now is to have this information alongside, say, clinician reports, and to, in the event where there's a significant discrepancy, I think it would be a step forward to pay attention to what the patient is saying on the PRO and factor that in somehow into decision making, rather than not having that information at all and not being able to use it in any way. So I think right now the emphasis is on capturing that information, at least having it available. And at least from my experience, I just don't have a good answer of how, algorithmically or systematically how to incorporate that and to say, here is exactly how we make a decision based on that new information.
Yeah, I can also add a little bit. I think, again, industry is still learning how to use PRO, especially in the early stage of the dose finding decisions. So I think the PRO has a lot of advantage, right? You collect more often, frequent, compare, you only visit clinical site maybe once a month or twice a month. But again, the other level, there's also nuances because there's integrate a lot of inter-subject variability. One patient may wait pain, very extremely pain, and the other maybe wait is okay. So I think there's a lot of subjectivity. We're still learning how to see, going through the noise and identify the real issues. I think FDA recently in the ASCO meeting to share that, to how to do the exposure response for the specific item, for example, diarrhea. So that one is less subjective. You can look into how many you need to go to bathroom per day so that they can count off the variability. So again, how to use PRO, we're still learning. And hopefully we don't generate additional confusion versus give us a little bit better feeling on the patient tolerabilities. But I think as an industry and academic, all together, we should learning together how we want to tackle this data. And the other side is related to your comments on the, how we dose, titration. So I do have some example. For example, Pfizer asatinib. Actually, that one because they have some hypertension, AE is more like mechanism related. So actually, they even titration up based on your AE. They start 5 mg for every patient. If you don't achieve some certain level, the AE hypertension, you are allowed to titration up. And if you see that AE, you also allowed to titration down. So I think this way, due to the high inter-subject variability for the PK, so when you do that titration based on the AE, you minimize the inter-subject variability for the PK in the end. So we do have some prior example how we use the AE or use the efficacy to titration the patient. And the other example is inavolisumab, the ADC for cleomycin. I think in there, they based on the response. In the AML, they can quickly achieve the CR after 1 cycle treatment. So based on the 1 cycle response rate, if the patient achieves CR, they can go to the lower dose. If we don't achieve CR, get a partial response, they continue the higher dose. So we do have some example, but in the case, we need to understand if in the case for the 2 titrations, we need to understand those response relationship. Maybe their therapy window is very narrow. They need to do that. Not necessarily every drug need to do that. Yeah.
Maybe I can comment on the PRO issue. As a clinician seeing patients on phase 1 trials every day, one of the issues for patients is they're dying of cancer. They're terrified the drug is not working. or they're terrified the doctor will take them off the drug that is working. So there is some underlying bias here with regards to toxicity reporting by the patient. If the patient thinks the drug is working and they're desperate to stay on drug, they are likely to underreport to the physician, and they may actually give more of the information about toxicity to the nurse. So we would regularly get the nurse to speak to the patient independently of the physician to try and get a more complete picture of the tolerability of the agent. But what I'm trying to get at is if the patient thinks they're benefiting and they don't want to come off trial for any reason or dose reduce, they're much more likely to not tell you the toxicity. And that's the underlying bias that we as humans, I guess, use to cope with the stresses we're going through. And I think that's very, irrational. And so I think we have to think about the EPROs quite carefully, because there will be conflicting data sets coming through.
Yeah, sure.
I completely agree. This is Sophie. I completely agree with what Johan just said, and just say as well as having worked in different countries in phase 1 trials, I can say that there is a cultural thing as well, which may completely bias the PROs reported by the patients, and also whether they have or not alternative treatment options afterwards. That's why I think we have to really think very carefully about the interpretation of the PROs. It's a super useful tool, but we just have to implement it alongside what we already have. Yeah.
Thank you, Sophie. One, and I'm afraid last question here from the audience. Jaap.
Good. CDDF has the last word.
Of course.
Yeah, for CDDF. Picking up on Johan's point, I mean, yes, patients will possibly underreport because they are biased to stay on drug. On the other hand, I bet you that physicians will certainly underreport, because that's also an element that we know. Now, if we take that into account and look at Sophie's data, we started that whole mission of trying to get chronic DLT definitions by stating, what if you would have nausea, grade 1, for every single day for the rest of your life? Would you take that drug? As a healthy human being, it's easier maybe to answer the question, and you might say, well, yes, but if you're sick, you might feel different. So patients' opinions are important. And I would strongly recommend to use PROs early on as possible, because I'll bet you they're not going to underreport. And that's important. We need to know that information. We need to know the phase 1 populations are different, Johan said that correctly, from the average population out there. If they are reporting something that is really serious to them, I'll bet you that the average population will consider it completely intolerable. So don't ignore the power of the patient-reported DLT and AE.
Thank you very much. I'm afraid we are at the end of this session. Thanks a lot again to all the speakers, to the panelists, and to a very lively and informative discussion. Thank you. And I would like to ask for session 5 for Katrin and Axel to join us here for the panel.